So there are several applications of polymerase chain reaction. It is widely used in the research as well as in the molecular diagnosis. In research, it has several applications in cloning of the gene, in vitro mutagenesis, sequencing and mutation identification, SNP genotyping, gene expression, etc. And some of the same applications are overlapping with the molecular diagnosis as well. For example, genotyping sometimes is used for identification of a specific mutation, and the same application we also use in the molecular diagnosis as well. So, talking specifically with the molecular diagnostic applications of the PCR, we can use the PCR in the DNA fingerprinting for the paternity testing, for the crime scene investigation. Uh, the PCR is also applicable to detect the infectious diseases, for example, HCV, HIV, and the newly emerged coronavirus infections, etc. And the PCR can also be used for the detection of specific mutations that are causing the specific genetic disorders, and whether it is the rare genetic conditions or the cancer conditions as well. So, a very specific application of the real-time PCR in the molecular diagnosis includes the probe-based detection for the presence or the absence of the infectious microbes or the viral particles. SNP genotyping, that is used for the detection of a specific mutation in a specific gene that is causing either a genetic disorder, inherited genetic disorder, or cancer condition. And the real-time PCR is also used for the copy number analysis, that is used for the detection of the loss or the gain of the DNA sequence, and is also responsible for certain kind of genetic disorder as well. So let's take a very famous example of detection of the coronavirus, SARS-CoV-2, that is causing the COVID-19 disorder. So generally what happens when you go to a lab, so they take your uh, oropharyngeal or nasopharyngeal sample because the viral population is pretty much high in that specific area as it is a respiratory virus. So when the sample is taken, it is the DNA, the RNA is extracted from the sample, and then after extraction of the RNA, the RNA is converted to the cDNA, and then the cDNA is then detected. So here is a specific example. Sample is collected, RNA is isolated from the sample, then RNA is reverse transcribed, and after the reverse transcription, then again the DNA polymerase it amplifies a specific targeted gene in the coronavirus. So the probe is then hydrolyzed, and after hydrolysis of the probe, the reporter dye it emits a specific fluorophores radiations. So if the virus is there inside the sample, obviously the probe it will bind over the target because the probe is a specific DNA fragment, DNA molecule which has complementarity with the target. So if the target is there, the probe will definitely bind with the target, and the DNA amplification will happen, and the probe will be chopped off, and the radiation will be emitted, and the patient will be detected as positive. And on the other hand, if the sample doesn't has any viral particle, then obviously the probe will never find its target. So as the probe will never find its target, obviously the reporter dye will not be able to emit the radiation, and hence the patient will be declared as negative for the SARS-CoV-2. Sansure Biotech China is one of the earlier launched molecular diagnostic kit for the SARS-CoV-2 detection. In this kit, they are providing the detection chemistry for ORF1ab gene and the N gene. The probe that is detecting ORF1ab gene is labeled with the FAM dye, and the probe that is detecting the N gene is labeled with the ROX. While for the internal control, the probe is labeled with the Psi5 dye. And here is given the thermocyclic condition for both the reverse transcription as well as for the cDNA amplification and the target binding of the primer probe. And here we goes for for the basic detection formula of the SARS-CoV-2. Actually, in the diagnosis, the CT value uh, is being checked for either the presence or the absence, and every kit has its own threshold values for the CTs. The CT is basically a cycle threshold value which shows that at which cycle the detection is started. So the next application of the real-time PCR is mutation detection in case of genetic disorder as well as cancer conditions. For that purpose, multiplex Stackman assay for the genotyping is performed, and all the probes specific for any specific SNP is used in a single reaction. And every probe is labeled with a different color dye. So upon the detection of a specific color dye, it specifies a specific SNP actually. So for example, if we take an example of a sequence change G to A, so two probes are will be used in the reaction. One probe will be specific for the G nucleotide and the other probe will be specific for the A nucleotide. For example, there are two probes. One probe specific for the G nucleotide is labeled with the Vic dye, and the other probe specific for the A nucleotide is labeled with the FAM dye, and both the FAM and the Vic dye they will emit a different radiation. So upon the proceed of the reaction, so if the detector detects the yellow color dye, for example, the Vic, then it will show that the sample contain the G nucleotide. The sample has the G nucleotide. And upon the emission of the blue color radiation, the blue color fluorescence, it will indicate that the sample has A nucleotide. So this is how the multiplex reaction it works. And for example, if the detector detects both color dyes, so it shows that uh, the person is heterozygous because on one chromosome is present G nucleotide, on the other chromosome is present A nucleotide.